All right, so today, so the next thing we will discuss is that how genetics can lead to cancer. So this is just to tell you what are these genetic alterations, right? Genetics can cause cancer. So you have a little bit, hey, so I tell everyone, no cell phone, okay? So do not use cell phones while you're in the class, okay? So what happens is that we are constantly exposed, even spontaneously, right? Our DNA gets damaged. So you are sitting in the class, you are not exposed to any bad thing in the class, but spontaneously DNA is getting damaged, and this is just simple chemistry. So you know the phosphodiester linkage, right? So the DNA is linked by phosphodiester linkage, but what happens, just simple chemistry inside the cell, the purine bases are removed. So the whole phosphodiester linkage is there, but purine base is removed. So it looks like, you know, you look at the teeth, right? So all the teeth are attached to the gum, right? And if you remove the tooth, the gum is still there, but the tooth is missing, right? So the DNA is still there, but the base is missing. So these, when purines are attached, they can just spontaneously get hydrolyzed. Okay, the glycosidic bond which is linked to the purine can get hydrolyzed and purine can leave. This process is called depurination. And this happens 5,000 times in every cell per day. So every single cell in our body undergoes depurination at least 5,000 times. The DNA undergoes depurination at least 5,000 times per day in every single cell. So it is immediately repaired. Or we can have some outside exposure. So similarly, there are many processes which can damage the DNA. Similarly, pyrimidines can also spontaneously hydrolyze, but not as much as purines. So there are DNA repair enzymes which can repair it. So you know DNA, so we have two copies of the DNA. So if one is damaged, a single base pair chain in one of the strand, then the other strand serves as a template and it can be easily repaired. Okay, so that doesn't cause bigger harm. But if both the strands are damaged, there are many ways to even fix both the strands there. Okay, so but many times what happens is that gene itself is amplified. So suppose you are supposed to have only one copy of the gene in the DNA, but there is the DNA itself undergoes amplification. Now you have many copies of that gene in the DNA itself. That is called gene amplification. Sometimes the gene is removed from the chromosome. Okay, that can actually, so P53, we all know P53 is a tumor suppressor. Many times P53 is mutated so that it is no longer functional. Many times P53 is altogether lost in cancer patients. Okay, so gene deletion can cause. Similarly, structure variations. I will show you the examples of all of these. Okay, so I have already shown you this slide. And I have already shown you that if there is a germline mutation in sperm or egg, then the child also inherits it. But if there is some mutation in some part of the body, in some tissue, that is that we have acquired due to the environment, we are not going to give it to the children. Okay? So this is showing you mutation in the germline in this case. So this will get in the embryo, and that embryo, when they develop, they have that disease, and they are also carriers of that disease. But if the mutation is not present in the germ cell and it appears later in life, even at the embryonic stage in some other tissue, they are not going to be the carriers of that disease. Okay? So this is showing you all different kinds of inherited cancer risk. Bone cancer can also be an inherited cancer thing. So basically every single cancer that is known, it has some genetic component. Some percentage of those kind of cancer can arise from genetic alterations. So now let's look at the mechanisms. What is happening? Okay, so I have made 4 different categories. Oncogenes. You know what are oncogenes? Oncogenes are genes that cause cancer. Okay, so oncogenes. So suppose you so a very famous example is SRC protein, SRC, SRC protein, right? So normal SRC does not cause cancer, but if there is some mutation in SRC and it is always on, always active, so normal SRC will become active for some time and then its activity will go away. But then a mutation can keep it active all the time and that will cause cancer. So SRC at that stage is called a proto-oncogene. So it has that capability of causing cancer. But in normal cells, when it is in the inactive form, it will not cause cancer. But if it is always active, then it will cause cancer. So why? Mutation can make a simple protein which is good, it can make it bad. Okay? Or amplification. Amplification means in the DNA itself, you have many copies of that gene. Or translocation. I will tell you the real example of translocation in just a few minutes. Tumor suppressor proteins are often lost or they become defective in cancer. Again, P53 is the best example. Okay, DNA repair enzymes, if they are missing, DNA cannot be repaired. And if DNA cannot be repaired, that is a very big cause which can lead to cancer, and then we'll discuss about epigenetic alterations. So I'm going to focus on these 4 things here. This deals with epigenetics. This is the tumor suppressor. These are the oncogenes and how genomic instability and mutation can lead to cancer. So remember I started the lecture from this slide. These are the 14 features of cancer. Okay. So oncogenes, okay. So this is a normal gene. I've written it here is an oncogene because what happens is that in normal cells it is active for some time and then it becomes inactive. So it responds. So it is like there is this person. So this person is told you become active. He's doing the thing, and he's taking care of that. And when he has to shut down, he's just stopping it. But then this in cancer, this does not respond to outside signal. This is running the show all by itself. So when it is asked, do not divide, it is still dividing. It just is independent of the outside signal. That's when it can lead to cancer. Okay. So suppose there is a protein. Suppose there is a cell, right? Let's assume this room is a cell. And let's assume for now that this is the nucleus where I'm standing, okay? And that is the cell surface. And I am the person. I am the oncogene here. So the signal comes, some growth factor comes, the signal is told, it is told to me. Then I tell the nucleus, and the nucleus then responds, and the cells divide, right? But I have to get that signal from there before I can tell the nucleus. But then there is something changes in me and I don't have to listen to that growth factor there. So now whether the growth factor is there or not, I am still active. So I will just give the signal, keep dividing, keep dividing, keep dividing, keep dividing. That's an oncogene because I don't have any shut-off mechanism anymore. I just do not respond to that. Okay. Okay, so this is, and why did I become like this? Because, presumably because of change in me, because I don't have to adhere to that. So that is the mutation. So if there is a mutation in the DNA, RNA will also have that kind of mutation, and now this protein will be different because there is a mutation. Do you guys all know what is a mutation? Every one of you? You also know, right? All of you, you also know what is a mutation, right? So that mutation can make me completely active all the time. I don't have to be. I can give you many specific examples, okay? So I will give you a real example. So this SRC protein I was telling you, So this Sark protein has a kinase domain, and ATP binds here and it is active. Okay. So when it is inactive, I'm just making a cartoon. When it is inactive, this active site is closed. I'm just making it like this. But when it is active, this active site is open. Okay, so if the enzyme active site is open, it can catalyze. If it is closed, it will not, this thing. So the mutation can keep it open all the time. Okay, and then it will be active all the time. Okay, so but in normal cases, this is closed. Why it is closed? Because there is some conformational change here which keeps it closed. When the growth factor signaling comes, this protein changes conformation, it opens up. But if you make a mutation and always keep it open, it doesn't need any outside signal. So that is what it is saying, mutation here. See, under normal circumstances, you should have one copy of the gene in the DNA, but now it is amplified, so you will always make more protein. So once again, I want to tell you, if many times our RNA is defective, because RNA is made from DNA, RNA polymerase makes the DNA, and many times it puts the wrong base because it's doing it fast. But if RNA is defective, no problem, because RNA has a short half-life, it will get degraded. If RNA is defective, it will make the wrong protein, and then protein itself will get degraded. Half of the time, if the protein is wrong, protein doesn't fold properly, and if it doesn't fold properly, the cells will immediately clean the misfolded protein. So it will not even damage us at all. Okay, but if DNA is damaged, and if DNA is mutated, every single time the same RNA will be made. And even if that RNA dies, some other RNA will have the same problem. So we can never get rid of that problem. That is why DNA damage is bad, RNA damage is not bad at all, right? So I can change my statement. I always say never in biology, in science. In science, you never say never, because there are always exceptions to every rule. So I take back that sentence that RNA damage can never be bad. RNA damage can also be bad, but DNA damage is worse. Okay? And then chromosomal rearrangement. Okay? So I will give you an example of this thing. Okay? So chromosomal rearrangement. Do you guys know what is a promoter of a gene? You don't know that promoter of a gene. That's okay. I just want to. So do you know, Leknath, what is a promoter? So let me give you a quick crash course in biology here. So DNA makes RNA, RNA makes protein, right? All of you know that part. Okay. So this is DNA. DNA makes RNA, and RNA makes protein. Okay. So our earlier, not right now. So we have 3 billion base pairs in humans, and people used to think that only 5% of that makes proteins. So we have 3 billion base pairs. So let's say that is our 100% DNA. So this is true that 5% of that 100% DNA makes protein. So what is happening with the 95%, right? So people used to call it, scientists used to call it junk DNA. If we cannot figure out something and we think we are very smart, we call it junk. So the scientists, and you go in the journals, they call it junk DNA. That 95% of human DNA is junk DNA, 5% is this. So whoever labeled this junk DNA really thought that nature was not as smart as that person, because nature has made 95% junk DNA. Now we know that is not true. This is something which has come in the last 10-15 years. Okay. So because I have myself taught in 2004 that 95% of human DNA is junk, because that's what we all believed in 2004-5. So that 95% also makes RNA, but what is still true is that they make RNA, and these RNAs have different roles, but these 95% are still not making proteins. So they are not also protein encoding DNA, but they are making RNA. You guys have heard of microRNA and all these things. So that they are coming from those, and they are called non-coding RNA because they don't make proteins. Okay. So then the 5% which makes protein, or all the RNA that is made, it's not 100% DNA. We can say safely that at least we don't know as yet that 100% DNA will make 100% RNA. Only small percentage will make. So how do cells know which RNA they should make and which RNAs they should not make, right? So DNA has this region called promoter. Okay. So when RNA is made, the enzyme which makes RNA is called RNA polymerase. RNA polymerase. So this RNA polymerase will recognize special sequences on the DNA which are called promoter. It will bind here and then it will make the RNA. That makes sense? So that means promoters are very important because they are the one which will tell this RNA should be made. Okay. So if the promoter region is hidden, RNA polymerase cannot access it, and that RNA will not be made. So the thing which is written here, enhancer. So these are the proteins which will expose the promoter region so that RNA polymerase can make RNA. So if it is super exposed in cancer, then you will make a lot more of this RNA, and if you make a lot more of this RNA, you will make a lot more of this protein, and that can be bad. So that is what happens in diseases. Something which is not supposed to be that exposed can get exposed, or for tumor suppressors it will get hidden. So it is RNA is not made. Is it making sense? So that can also lead to abnormal protein expression. Okay. So here this proto-oncogene means this gene has the ability to act as an oncogene if it is overexpressed. Okay. So there is some mutation, now the protein is abnormal. Or see, this is what I was telling you. This, let's say it is the promoter region. There is something in there. So now you make a lot more protein. You are not supposed to make it, but something happened here, so it is more exposed, more protein. Okay. So here there is, okay, here we have added either a new promoter, which is very strong promoter. So now, so you know, not all promoters are equal. Okay, some are strong, some are weak. So suppose you open up a cell. Have you guys heard of proteomics, 2D gels? You have heard of it. So I used to show a picture of a 2D gel. If you have ever seen a picture of a 2D gel on the web, have you seen that? Have you seen any 2D gel picture? Anyone? Okay, so suppose every protein appears as a dot, right? The beauty is this, that if it was equal, if all the proteins were equal, every dot will be equal in size, right? But every dot is unique. Some is big, some is small, right? So you guys agree with me, not all proteins are expressed at equal levels in the cell. Some are present in very small amounts, some are present in a large amount, right? And it has to be that way because some proteins are so powerful that they're expressed in very small amount for a very short period of time, and they do their job and they're immediately degraded. And these proteins, if they stick around for a long time, that's a very bad thing. Okay, so what happens is that here something happens, and this makes it very strong promoter. So a lot more protein is produced. That's bad. That can lead to, because this is a proto-oncogene. This has the tendency to become cancerous if it is present in huge amount. And see, all of these are leading to huge protein production. So now it becomes an oncogene. Some people have inherent quality to become bad, right? If you keep them in check, they are okay. But if you give them too much power, that is bad. That's the proto-oncogene. Okay? So see, gene amplification, huge amount of protein, cancer. Okay? So that was oncogene. Now tumor suppressor genes are the good guys. They keep tumor in check. So here what you want to do, we don't want to do, but cancer, if they are lost, cancer develops. Okay, so here there are many tumor suppressor genes. So when these tumor suppressor genes are there, they keep the cells from dividing too much, and because of that cancer never develops. But if they disappear, then if they are defective, they are there but they are not able to do their job, cancer will develop. Okay, or this is let's stick with we'll not go to the oncogene, we'll just look at the tumor suppressor. So either they get deleted or they are defective, and that is a root cause for cancer. Okay, so here a tumor suppressor gene is there, and some kind of outside environment, like even second-hand smoke. Okay, second-hand smoke or radiation or UV, this thing. Now the tumor suppressor gene, TSG stands for tumor suppressor genes. So because of that there is a mutation, they are not functioning, and cancer, it becomes cancer. P53 is, as I said, the most famous example. So P53 is lost or defective in many different kinds of cancers. Okay. So then let's come to DNA repair gel. So our DNA is getting damaged all the time, and it is getting repaired also all the time. Unless we expose ourselves to really severe conditions where DNA is damaged. So right now we are here, and in our cells depurination is occurring. That's a normal thing. It can be repaired, but if we go in the beach and we sit there without any sunscreen for hours and hours, the damage can become so extensive that the body's repair system is so overwhelmed that it cannot repair it completely. Okay, and that can lead to disease. Okay, so accumulated DNA damage. If it dies, good news, right? But if it doesn't die, then the cells become very aggressive. If stress is there and if they die, that is good. But if they just keep on taking the stress and they modify themselves, then that is when a disease develops. Okay. So this is here. So it shows you type of DNA damage which is occurring in our body under normal circumstances, and this is all normal processes which is occurring. So we can depurination, the purine is hydrolyzed. It can be simple depyrimidation, also pyrimidine is also hydrolyzed. Cytosine, you guys know, has an NH2 group. NH2 group can get hydrolyzed to oxo group, keto group, so deamination. Single-stranded breaks, not big deal, because the second strand will act as a template, it will be fixed. Base oxidation, guanine has a hydroxyl group, it can be fixed. This is happening all the time. Every time we are going out in the sun for a long time, even taking long walk in the sun, thymine will form cross linkage like this, or cytosine can form cross linkage, or cytosine and thymine can form cross linkage. But then this light, this simple visible light, not sunlight, simple visible light will reverse that. It will be, that is the easiest fix. So this is not damaging at all. And then sometimes DNA repair enzymes can put the wrong base. It can be fixed, but if it is not fixed, it can lead to mutation. Okay, base alkalization. So these are many different kinds of defects which are being repaired in our body. But if we are constantly exposed to this thing, that's when body gives up, and the body cannot repair this thing. And if it is not repaired, it can give rise to cancer. Okay, so and diet comes everywhere. Okay. So these kind of problems can give rise to DNA damage, and although the body repairs it, our exercise and diet, these two are big things which can prevent DNA damage from DNA damage. Okay, so this is the antioxidant defense system, DNA repair system, and usually body has this safe mechanism that if it cannot repair it, the cells will die. It will turn on the warning signal for cell death, and the cell will die. But then if the cells resist death, those are the cells which are the dangerous cells. Okay. Now let's talk about BRCA1. You have heard of BRCA1 since yesterday. So what exactly happens in BRCA1? So BRCA1 and BRCA2, they are DNA repair enzymes. And they are so famous because they are often deleted in cancer. Okay. So if DNA is damaged, BRCA1 can repair it. But if BRCA1 is absent, DNA damage goes unrepaired. And so this BRCA1, BRCA2, if they are defective or they are absent, it can lead to many different diseases. The most famous one are breast cancer and ovarian cancer. Okay, so that was about the DNA repair enzymes. The last thing is the epigenetic reprogramming. So do you guys understand epigenetics? I told about it a little bit yesterday. I'm going to tell you about a lot more today in the lectures. But epigenetics is that we do not change the DNA. So I will tell you something. Some of you guys know about Alzheimer's disease, right? So in Alzheimer's disease, patients lose their memory, right? More than the age-related memory. So when people get older, some people, not all, some people, they have age-related memory loss. So they often say, oh, I cannot remember this or I cannot remember that. So that is not a big deal. That is just age-related loss. But few people lose memory at a much rapid rate, okay? And that is most likely due to Alzheimer's disease. But even those Alzheimer's patients, if you know of any such cases, Alzheimer's patient will remember their children. Okay, they will remember their children. So Ronald Reagan, obviously you know Ronald Reagan, all of you. So he had Alzheimer's disease. Up until his last day, he did not forget his wife. He was so happy all the time to see his wife. He never not recognized his wife. But all the other people, he forgot that he was the President of the United States. He forgot all his colleagues. He forgot, but he did not forget his wife and his children. So these people have long-term memories. All the things they did in their childhood, they will tell you all the childhood things. Okay, they will tell you all about their old thing, but they would not be able to tell that, you know, whatever story you shared with them yesterday or today. Short-term memory is lost. So people were very curious why, even in many cases, why people can recall long-term memories and why they cannot recall something which happened yesterday, day before yesterday, even a year ago. So then I was in a conference and they presented a paper many, many years ago. It is due to epigenetic modifications. So the long-term memories, the DNA has epigenetic changes and they make the long-term memories. And those epigenetic changes do not occur in short-term memory. And we cannot recall those things many times. Like many of us, I mean, you guys are very young, but the childhood stories we still remember. But what we did yesterday or the trivial things of yesterday we have no recollection of those things. So that is also due to epigenetics. So it is very important. So the gene is there, but epigenetics decides whether the gene is going to be expressed to make proteins or not. So that is why epigenetic modification. So it's not surprising. Cancer hijacks the epigenetic machinery to express the proteins it wants to be expressed, or to suppress the expression of good proteins and increase the expression of bad proteins. Okay, so now let's talk about what exactly is epigenetics. Okay, so so if we take all the DNA from our body and from one chromosome we take the DNA, and chromosome is what? Highly condensed DNA along with proteins. So chromosome is made of two, we know our DNA is stored in the form of chromosomes. So chromosomes is made of two components, DNA plus proteins. And this DNA, so we have problem in really cleaning our house, right? So many times we clean it properly, many times we even dump things when we are in a hurry, right? cells cannot dump DNA. If you take a chromosome and you unravel it, so it's very nicely packed. If you unravel it, one single chromosome DNA can extend to 2 meters, and we have 23 chromosomes. So that much amount of DNA is present in the form of chromosome, and it is packed inside the nucleus of a cell that we cannot see with our eyes. So in a very, very small area, that much DNA is there. But the cells do not have a choice of aggregating all the DNA and just making it a compact ball and just putting it in the nucleus. It is highly organized. Why? Because our cells are dividing all the time. So the DNA needs to be replicated 100% almost every 2-3 days. 100%. So the 100% of DNA has to open up, get replicated. That's one thing. But the same DNA is making RNA, right? And RNA is being made every second in our body. So, and it has to decide which RNAs to make. So DNA is opening up, closing, opening up, closing. So it's an extremely organized structure. You cannot just aggregate the DNA and just make it small just to fit in the nucleus. It's nothing like that. It's a highly organized structure. So this is what it is showing you here. This is chromosome. You open, this is here. It appears that this is just this. No, there are huge level of organization here, huge level of organization. I'll show you different pictures. But when you open it, after a lot of organization, you will come to this kind of structure. And each of these are histone octamers. So DNA is negatively charged because of the phosphodiester linkage. And these proteins have lysine and arginine, so they are positively charged. So this is an octamer of histone protein, and DNA wraps around it. So it's like you are wearing something with beads on it, okay? So it looks like beads there. And then you open up all the DNA, you unravel it, and you will see the DNA like this, okay? So this is highly organized structure. So this is chromatin here. So some part of chromatin are more open, so those DNAs are more open, some are highly compact. When DNA gets replicated, no matter what, DNA is always replicated 100%. It cannot be 99%. If it was 99%, we would not live to the age we live up to. DNA has to be replicated 100%. Okay, but some part are more open than the other. So those parts which are more open, those are the part which makes more RNA. The parts which are closed, they don't make those many RNAs. They are replicated, they are still replicated, but they don't make as many RNAs. And that is, so what decides that some part of the DNA should be closed, some part should be more open, and that's where epigenetics comes into play. So DNAs, they undergo modification. The most common modification is methylation of DNA. So this is cytosine, and cytosine can get methylated at this position here. I will tell you the consequence of methylation. So once the DNA is synthesized, you know, when when you have cytosine, guanine, cytosine, guanine, cytosine, guanine, cytosine, guanine, so if you have too many CG, those C's, not the G's, they have a tendency to get undergo methylation. So when they are methylated, so then they form 7-methylcytosine. So you know the sequence we read. If cytosine is followed by G, we call it CG. But long time ago, maybe 50, 60 years ago, people used to call it not CG but CPG. Why they call CPG? P is the phosphodiester linkage, which is telling you that they're linked by this thing. You know, like, you know, when I was growing up, we started abbreviating a lot of things, right? Now you guys are abbreviating even the abbreviated version, right? So I have to say, oh, that's what you mean. Right now, nobody says billion. It's too hard to say billion, so they call it bill. The next generation will not even call bill. It will be more abbreviated. We cannot even think what they are going to call. So you know, it's getting abbreviation. Our abbreviation itself are getting abbreviated, right? So those time people were calling CPG, now we call it CG because that's understood. Of course, it is a phosphodiester linkage. But if you look at the textbook, it will say CPG, CPG, CPG. Those are CG, CG, CG. And they are the one which are getting methylated. So I'm just telling you the most common modification. So there is methylation, there is acetylation, and deacetylation. So remember, there are proteins, histone proteins, and DNA is wrapped around it. So the most common changes in DNA is methylation. So arginine and lysine residues in DNA, they can get methylated. For histones, they can get methylated also, they can get acetylated also. Acetyl group is added. And then the acetyl group is also removed here. So this is lysine side chain, and this lysine is acetylated. But lysine can also be methylated. Okay, so look at the colors here. So I have made acetylation green because acetylation opens up the DNA, and DNA is more accessible. Methylation closes the DNA. Okay, so acetylation, it opens. Acetylation occurs not in the DNA. Acetylation occurs in the histones, and DNA will be more exposed. Okay, I will tell you more in details in just a minute. Look at the signal here. So here this acetylation is not occurring in the DNA, it is occurring on the histone protein. And look at this, this is a green signal. So see, I just told you about the promoter region. So when it is acetylated, the structure is more open, and promoter region is this one, it is C exposed here. So now RNA polymerase can bind here, and the RNA will be made. But here it is acetylated, so this is the normal form. When it is acylated, it opens up. Okay, but when it is methylated, see how close it is. It's even more compact, because once it is methylated, these proteins come and bind and completely close the DNA. Now RNA polymerase cannot reach there, and these DNAs will not make RNA. This part of the DNA will not make RNA. So methylation is red signal, acetylation is green signal. Okay, so look at this thing. So it is showing you two parts, protein part, DNA part. When DNA is methylated, it is closed DNA. That does not make RNA. Okay, when it is not methylated, it makes RNA. Histone, when it is less acetylated, less accessible. When it is acetylated, more accessible. Okay, so because of that, so what? So now you can guess yourself. If I have to ask you, I'm supposed to ask you questions during tutorial, but I'll ask you now. So suppose cancer has to play with epigenetics. What will it do to the oncogenes in terms of epigenetics? Tell me that. Anyone? Of what genes? Acetylation of which kind of genes? Oncogenes. Very good. It will do acetylation of oncogenes because then it will be more expressed, more RNA, more protein. Very good. What else? What other kind of things? Methylation of tumor suppressors, right? Tumor suppressors are not going to be expressed. So now you know exactly what they are going to do. So it is not surprising at all that epigenetics plays a very important role, and cancer also hijacks the epigenetic machinery. Okay, because those are the ones which are telling which ones to be expressed and which ones should not be expressed. Okay, so that's what it is telling you. So, so this is epigenetic problem. What you said is now shown here. So normal cells. So now you guys can follow too. Promoter region. Can you see promoter target gene? So this guy is methylated here. So methylation will close it, correct? So this will be, so for, yeah, so this is methylated. So then the target is not expressed as much. So in normal cells, oncogenes are off. But in cancer, no methylation. Now this is expressed in huge amount. Is it making sense to all of you? Now let's look in terms here. Okay. So in this case, in normal cells, it is partly methylated. So that means it is partially exposed and it is present in small amount, right? Partially exposed. But now, in this case, hypomethylation, barely any methylation, too open, a lot more gene is expressed here. Do you guys know what are exons and introns? You know? You don't know? That's okay. We'll discuss that later. Right now, just treat them as genes. I'll come to exons and introns later. Okay. Now here, in this case, let's assume this is a tumor suppressor. So in tumor suppressor, they will hypermethylate it. So the tumor suppressor is not expressed. Okay. And this is a normal circumstances. So, okay. So in this case, so every time you see here, right? So this is one gene, this is another gene, this is another gene. Okay. So these are three different genes. Okay, so don't worry about exon or intron right now, just treat them as a gene here. And every gene has a promoter. Okay, so the promoter for this one is this one is this one is this, and for this, these are the promoter region. For this exon, you have this promoter, for this exon, you have this promoter. So look at the methylation level. So some are hypermethylated, some are hypomethylated. And accordingly, those genes which are hypermethylated are not going to be expressed. Those which are hypomethylated will be more expressed. So just in nutshell, oncogenes, hypomethylation. Tumor suppression, hypermethylation. Okay. So, so this is telling you about the clinical relevance of all different kinds of things. Whether your tumor suppressors are deleted, whether they are functional, you can check it by genetic testing. Yes, go ahead.
What is the mechanism behind that methylation causes the contraction of the aspiration causes the
So there are many DNA methylases. Okay. So some DNA methylases are overactive or less active depending on that. So they have substrate specificity. So not all methylases will methylate all promoters. They will do in specific context. So those DNA methylases, if they are overproduced, which methylate or they are absent in so those DNA methylase enzymes which methylate tumor suppressor promoters, if they are absent, if they are overexpressed, then they will hypermethylate those promoters, and those tumor suppressors will not be expressed, and that can lead to cancer. So basically, it is the expression level of DNA methylases themselves that can regulate. Second thing is that DNA methylation can only occur when there is some promoter exposed there, right? So there could be other mechanism where the promoter itself is not that exposed there. It is partially exposed, or there is something which is blocking the DNA methylase to come and hypermethylate. So it's a very, very complicated process. So it is not that all the promoters are exposed all the time where DNA methylase can come and methylate it or not methylate it. So there could be some kind of blockage here also which can prevent DNA methylase to come here and methylate this thing. So many factors can lead to change in epigenetics. Histones, yes. Repressed, yeah. Yes, yes, yes. Oh, that's so. Here I have only those slides which I brought it with me, so I don't have all the things, but I have really beautiful, beautiful pictures, in my lectures. So this is a very interesting thing, and I really wish I had those images here. So what happens is that, so you know the process by which DNA makes RNA. What is that process called? Transcription, right? Have you guys looked at the transcriptional machinery in humans? Probably not. Have you looked at it? So I am telling you a very simplified version. RNA polymerase makes RNA, which is 100% true, but it is a very complex process in the sense because DNA is so compact with all this chromosome kind of thing, right? So in human, the DNA is never open all the time, never ever. It just opens partially. So there are these many proteins, they come together, many proteins, they come together to keep the DNA, to expose the DNA. So what happens is that, think of the DNA like it, just think for it, that the DNA is extending from that wall to that wall here, right? So, and where I am standing, I am the promoter region, and this gene needs to be expressed, right? So you would think, because, well, the DNA is extending up until that, but I am the promoter region and this DNA is going that way. So you would think the whole show has to happen here because I am the promoter region. And these guys will have no say to that, right? Because this DNA is, we are part of the same thing. But this is the gene, this is the promoter region. So that DNA. But what happens in reality, because DNA is so tightly wrapped around histones and all of these things, there are proteins. What they will do, they will bind that part of the DNA. So on one end, so I am DNA, this is DNA, I am not a protein, but some protein will come, it will bind where that lectin is there, it will bind there, and then it will tilt the DNA and the other part will bind to this one here. So now it has distorted the DNA like this so that this part is more open. So what my point here is that there are hundreds of proteins which come together and bind different parts of the DNA to control the gene expression there. So if you delete something there, or if something is not binding, or if something is overexpressed, this gene will be more exposed, less exposed, or RNA polymerase. So RNA polymerase, so if this is the DNA sequence and this is RNA polymerase, it will not bind like this only. It has two legs, and these two legs are fixed legs. They are not like our legs that we can have two legs like this or this, right? We have some capability of moving, but it has two legs and it is fixed. So when it sits on the promoter region, this is fixed length. It's not like movable length. So it recognizes two parts of the promoter where its legs are touching it, right? So if the promoter sequences are changed at two parts, it is not going to bind very nicely. So remember I just said a few minutes ago that promoters are strong or weak. So intentionally. So some promoters are strong, RNA polymerase will bind very tightly, very happily, it will make tons of RNA. Some promoters are weak, it's wobbly, okay, it binds but not very happy, less RNA, right? So the main thing is that it is intentional, the transcription is not the same for all the genes. Some genes make a lot more RNA, lot more protein. Some genes make a lot less RNA and less protein, okay? And there are many proteins which come together, and one of the mechanism is, so epigenetics we are talking about. So suppose it is methylated, then many proteins will come and bind the methylated part. So remember I showed you here for DNA. Now here it is for histone. When it is acetylated, nothing binds here, more open. See how open it is now. But when it is methylated, these HP1 proteins come and bind like this and completely block access to the DNA. So acetylation can also recruit transcriptional machinery to help in transcription. So it's not binding this thing, it is actually binding to those proteins which aid in transcription. Makes sense? So keep in mind, I am showing you the simple version of everything, okay? Most of the time acetylation opens up, but once in a while acetylation can also inhibit it. But I am just telling you what happens most often, okay? All right, so now I'm, go ahead, yeah, go ahead.
Methylation is limited to lysine and adenine alone? some amino acids like glutamine or asparagine, they have in it too.
They have NH2. I have never seen methylation there. It's mainly limited to arginine and lysine. I have never come across glutamine or any other amino acid besides these two that are methylated. And lysine methylation is most common. Okay, so now you guys already understand precision medicine. Precision medicine is that you find out exactly what went wrong with this particular patient and then you treat that problem. So suppose in many cases the main region for cancer is aurora kinase is deregulated. So you don't have to give chemotherapy, you just give aurora inhibitor to that patient, right? And that will be very good. It is besides the point that aurora inhibitor is also very toxic to start with. It is almost as worse as chemotherapy because aurora is expressed in every cell of our body, and if you inhibit aurora, every cell of our body suffers the consequence. But the point here is that if you figure out the exact mechanism, you can give that disease, you can give that therapy. So that is personalized medicine or precision medicine. So patient A has this mutation, so you give this drug. Patient B has this mutation, you give a different drug to that. That is precision medicine, right? So that is what it is telling you, that you don't, it is not one size fits all. Every patient is unique and every patient has to be given accordingly. See, many cancers which are very bad, they are caused by one major player, and if you find that major player, you just give that drug. Okay, so this is, now I'm telling you specific case study. So this is breast cancer. So a lot of breast cancer, they overexpress a receptor called HER2. Okay, so HER2 is also present in normal cells, and it is a growth factor, so it gives the signal to cells to divide, and when it is turned off, it is turned off. But in cancer, it is overexpressed. Okay, so because there are too many of them, too much signal is coming to the cells, and these cells just keep on growing all the time. So we know what is the culprit. HER2 is the culprit, right? Once we know, so this is one of the most famous drug. My friend took this drug, Herceptin. Her breast cancer is completely cured. She had breast cancer when she was, I don't know, she was in her 20s, and she was treated, and now she is very, for decades, no cancer. It never came back. So it's a very effective medicine. Okay, so Herceptin is an antibody against this HER2. So if you give Herceptin, no signaling. It just binds, keep them apart, and it is very effective. Breast cancer is one of those cancer which is very easy to treat unless it is triple negative. Okay, so breast cancer express 3 different kinds of receptors. So if it is a receptor associated disease, it's easy to treat because you give the antibody. Triple negative means all the 3 receptors are gone. And then how do you give an antibody? So that is the one which is very hard to treat. Okay. So this is lung cancer. So EGFR is also a receptor which belongs to the HER2 family. So what happens is that under normal cell conditions, when EGFR is so do you know what is a ligand versus the receptor? Do you guys know that? So do you know chemistry background? Probably not. Okay, you know. So let me still say it one time. This is a receptor, and this is so this receptor is poking out of the cell surface, right? So if this binds the ligand, this is the ligand. If it binds the ligand, it is turned on. As long as the ligand is not there, it will not turn on. Most of the times, these receptors are like this, right? And when it binds the ligand is like this. Okay, so it brings 2 receptors close, and when they are close, they phosphorylate each other and they are turned on. So in many cases, when they are separate, no signal. But this ligand is the one which will bring 2 receptors together. They dimerize, and once they dimerize, that's when signal is turned on. But when antibody binds, it binds separately. They will never come close together because now the antibody is blocking the binding of the ligand. So they stay apart, signal is off, right? So this is showing you different ways EGFR receptor is activated, especially in cancer. So under normal circumstances, they are separate. When EGF comes, two receptors come, signaling is turned on. EGF is removed, signaling is turned off, okay? But here what is happening, there is some mutation in there that even these are super acceptors. So super acceptors, you know, binding. EGF, when it binds, and if you have very little EGF and EGF binding is poor, then you will have less dimerization. But yesterday we were discussing, I don't know who was talking to me about prostate cancer and androgen signaling. I spoke to so many students. Somebody was talking to me about prostate cancer and androgen signaling. So one of the things they do, they increase their affinity towards the ligand. So here, even if you have less EGF, they are super binders, super acceptors. So even with less EGF, they will dimerize and turn on the signaling. That's bad. Okay, here, so remember I said two receptors come together, they phosphorylate each other. So here, they are always bound to ATP. This part is super active. So even if it doesn't dimerize properly, this part is already active and it is turning on the signal. So that is bad. And here, this is the extreme case. They come together even in the absence of EGF. They dimerize without EGF. So they are always turned on. They don't even need the outside signal. Okay, so what is the cure for that? You make, so many times the receptors, this pocket is like this under normal circumstances, but the pocket is mutated in cancer. So if you make an antibody which will recognize this pocket and not this pocket, so normal cells are unaffected, but the cancerous cells which are expressing this kind of one, they are the one which will be shut off. So this is what is mutate specific antibody for cancer only, okay? In this case, suppose it is a normal EGFR in cancer, but it is present in too much amount, then you can make an antibody and it will bind those and it will shut it off. Okay? Third case, CML. This is I wanted to tell you for a very long time. Okay? How many of you have heard of Gleevec? No one. So there is no chance that you have heard of this protein called BCR-ABL. So why Gleevec is so famous, and I'm part of that story. So Gleevec was the first tyrosine kinase inhibitor which got approved, and it got approved by Novartis. Okay, so and I was working at Novartis, and I was working in the kinase field. Having said all of that, I have nothing to do with Gleevec. But I was there when Gleevec was approved. So I was part of the celebration of Gleevec by Novartis. So I enjoyed all the perks without contributing to anything towards Gleevec. Okay, so we were flown first class to Mallorca in Spain, which is a very, very beautiful place on the Mediterranean Sea. And you know, we took a ship and that sailed on Mediterranean for almost a day. And throughout the day, we were served snacks. So they have a special word called tapas or something, I can't remember, but it's a snack on a stick. And all throughout the day, they were changing the snack. And it was huge celebration on the Mediterranean for Gleevec. And I did not do anything, but I was just there at the right time. So I had all the fun that originated from Gleevec. So it's my favorite story. Okay, so what happened and how it became a billion-dollar drug and saved the lives of millions of people. So what happens is that there is a kinase called ABL. Okay, so in normal cases it is off and it is turned off for a very short period of time and then it is turned off, right? So this is ABL. So this is inactive. So this is the catalytic domain which accepts ATP and phosphorylates protein. And this is off. So this is the whole protein. This is not different protein. One protein, different parts. Like this is head, this is torso, these are legs, right? So this is the enzymatic side. This is other domains. And the tail binds to this and keeps it off. And it is turned on for a short time, then it is turned on. What happens in CML, chronic myeloid leukemia, These patients have gene translocation. Remember, there was gene translocation. I said I'll tell you later on. So what happens is that one part of the other chromosome migrates and binds. This is ABL gene. So it migrates and comes here. I have another slide also I'll show you. So because of this fusion, ABL is a small protein, but this BCR from this thing translocates here. Now it becomes BCR ABL. And when it is in the BCR ABL form, it's always active. And that leads to CML. It's a very bad disease. Okay. Oh, this picture is already here. So see, ABL is this blue part here. Okay, and in BCR ABL, another chromosome, this BCR part comes from different chromosomes. So this is chromosome 22, ABL is in chromosome 9. But there is a chromosomal break where this part comes and fuses with this part, and it makes a huge protein. This is 220 kilodalton protein. And when this protein is expressed, ABL, the catalytic domain here is always, look at this thing, this is always, always, always on. And that leads to cancer CML. So this drug Gleevec, right? This Gleevec, so that binds exactly in the catalytic domain and shuts it off. And basically ATP can no longer bind and it is rendered ineffective. Okay, so this is how it works. And the other famous name for Gleevec, initially it was called imatinib, but the commercial name for patients who buy, that's Gleevec. So that is that story. So in many cases, something becomes hyperactive, and if you can design a drug which specifically binds that hyperactive protein, you shut it off. And another story about Gleevec is that, Gleevec was very effective for BCR ABL. So many companies made inhibitor which was even tighter binding than Gleevec, but it was not very effective in patients. So that's when it was realized that Gleevec not only binds BCR ABL, it binds PDGF receptor, c-KIT, and a few others, and they are all deregulated in CML. So that is why Gleevec is hitting all the bad targets in cancer along with BCR ABL, and that is why it is a relatively dirty drug, but all the correct targets, and that is why it is so effective in CML. But guess what? So when Gleevec was given to patient, it was fantastic. A large percentage of patients responded, and it was so much celebration. But then few patients in no time stopped responding. So then it was checked why they are not responding. So their gene was isolated BCR ABL. It underwent mutation in the active site. Now Gleevec no longer binds there. So that's the problem. They develop resistance because they undergo mutation. So this is the worst part of cancer. So I always tell when somebody is going chemotherapy, you better get it cured fast. I mean, it's not in someone's hand, it's not in my hand or that person's hand. But if the therapy continues for a long, long time, then the chances are cancer will develop resistance. That's why many people get well and then cancer comes back. That's the main reason, because cancer undergoes mutation and they become resistant. I'll stop here, right?